So I'm next. My name is Dr. Julie Seacombe. I have a lab here at Einstein. Firstly, I'd like to say a huge thank you, first of all, to Hayden from my lab. He actually was the one who reached out to a number of families through a Facebook page, and we had the most amazing morning this morning, talking to families that have been affected by KDM5C, and it was great to meet everyone, and I feel like it was just this really great interaction. And you guys, wherever you are dispersed around the place, you guys are the reason that we get up every day and do our research. Like, ultimately, we want to help. And, I'm going to tell you our little piece of that puzzle. And so, as this title says, we use Drosophila. And so by the end of this, if nothing else, I hope I've convinced you that cute little fruit flies are the key to everything. Okay, and so specifically, we're going to be talking about genetic alterations to KDM5C. Okay, so KDM5C is actually a gene that's encoded on the X chromosome. I'm not really going to dwell too much upon that. I'm going to say that, if you look through the literature, there are 31 different mutations that have been found in KDM5C. From talking to people this morning, there's a lot more that we don't know about KDM5C. If you look at the literature, you would see that it's mostly boys that are affected. Whereas if you talk to people, you talk to families, and you realize that actually there's a lot of girls that are affected as well. So even though the mutation is on the X, we still have a lot more to understand about this. And really, these are the range of severities that can happen. Mostly there's intellectual disability, but there's a whole lot of other syndromic features, which I'm going to come back and talk about a little bit more. So before I get into the details of KDM5C, I'm going to tell you that it regulates transcription. And I'm going to tell you that transcription is just fundamental to life on Earth. And the reason for that is that when you think about, we all start from a single fertilized egg, and in order for all of our cells to know what they're going to become, the stem cell has to activate the right set of genes. You have to activate the right set of genes, you have to turn the wrong set of genes off. And so fundamentally, you need to be able to turn on the right gene expression programs to become the right cell type. And then you need to maintain specific cell gene regulatory programs in order to maintain those cell types as well. And key to all of that is actually a group of proteins called transcription factors. And what these proteins actually do is that they bind to DNA. These proteins bind to DNA and they regulate the expression of genes. So next I'm going to tell you that obviously you all know that you have a lot of DNA in all of your cells. You have about 2 meters in every cell. And I'm going to tell you that in order to get that amount of DNA into your cell, you need to compact it. And one of the ways that this happens is by wrapping it around these histone proteins. So when you're talking about turning genes off, genes on, this all occurs in this context called chromosome, which is really DNA plus these histone proteins. And the reason I'm telling you this is that not only do these histones compact the DNA so that it can physically fit inside your nucleus, they also actively participate in whether a gene is able to be turned on or turned off. So when you do the really, really big picture stuff about what chromatin can do, when you think about a nucleus, you can think about the fact that there are some parts of the DNA. So in these pictures, the orange bits are the DNA and the gray bits are the histones. So there are some parts of your DNA that are just so compact and wrapped up in these histones that it's just inaccessible, right? The transcription factors can't bind to the DNA. In general, you can't activate those genes in those regions. And then there are other parts of your DNA that are actually more accessible. And actually regulating the transition between these two things is really important, because you can imagine that, so for example, actually what I meant to say was these is actually regulated by specific modifications. So these histone proteins have little bits of them that stick out from here. And for example, this acetylation mark actually affects the ability of this to form this compact DNA. So you get an acetylation group being added, and now you can open up the chromatin. And what this does is actually allow things like transcription factors to see their binding site on DNA. So obviously that's really important. The other thing that can happen is a different kind of mark could be added, and this is called methylation. And this is actually what KDM5, which is actually lysine demethylase 5. This is involved in regulating these methyl marks. And these methyl marks don't really impact the compaction of the DNA. What they do is basically form landing sites. So other proteins come along and they say, oh, I like this methyl mark, and they will come along and bind. And in the case of heterochromatin, very specific methyl marks actually maintain this compaction. And they actually allow this compaction to spread over greater distances as well. Whereas in regions that are generally more accessible, different proteins come in and recognize a different methyl mark, and these both maintain this kind of open chromatin structure, and they also allow other proteins to come in and to facilitate gene activation. So the reason I'm kind of emphasizing both the DNA binding component of this and the kind of chromatin aspect of this is that KDM5C is complicated. And one of the reasons it's complicated is because it does many things. So this is actually what KDM5C looks like. It's a little bit of a jumble. It looks like a little bit of a tangled bit of string here. And so the way that we like to kind of portray things as scientists is more like this. And the reason for that is it allows us to kind of isolate specific regions of proteins that do very specific functions. And so, for example, this arrowed domain here is actually this region down here of the protein that's actually contacting the DNA. So this is just a way for us to kind of categorize different bits of the protein that do very specific functions. Okay, so for example, yes, KDM5C can actually bind to DNA. It can also, one of the most characterized functions of KDM5C is actually this ability to demethylate. So this is actually mediated by two different regions of the protein that, even though in this amino acid string, they're kind of distant from each other, when you look at them in the crystal structure, they actually, they work right next to each other. And so what these different domains do is actually remove a methyl mark. So what you can imagine is that we know that it binds DNA. And the second thing we know is that it removes a methyl mark. So it's going to be affecting the way that other proteins can be recruited to chromatin. And of all of these different regions of the protein here, I've now described 3 of them. So there's a lot there that we don't know. Okay, so I'd also like to really emphasize that. We know some things, but there's also a lot that we just don't know about KDM5. I started working on KDM5, I think, back in 2005. Back then, we had no idea what it does. And so it's actually wonderful that we're actually starting to identify the things that it does do. So what about the mutations that are found in patients? So there's actually, I think I said there was 31 different mutations. And these can kind of fall broadly into 2 classes. So there's a number of mutations that actually, it doesn't really matter what these are. All the ones on the bottom here are expected to just remove the gene function altogether. So that's removing all of the different functions that KDM5C can do. Whereas all these different residues that I've shown on the top here are actually changes of 1 base pair of DNA, basically. And what that does is actually change 1 amino acid. So it's just in some ways really surprising that this would have dramatic consequences, but it's actually telling us something about what the protein is doing and that these residues are actually important. I'd also like to say that one thing that's, I'll touch upon in the next slide too, is one of the things that's a little bit frustrating for me from a genetics perspective is that one of the things that you might want to see from this is that knowing where your mutation is might tell you something about like the clinical outcomes that you might be able to predict. And this so far is just not true. In terms of you can pick any of the mutations down here that would be predicted to cause just loss of KDM5C, and you see a whole spectrum of different clinical outcomes from that. It's also true that you could imagine potentially that mutations in one particular region of the protein might cause a particular clinical outcome. And this is also just not true. And so I think, unfortunately right now, is that this makes it really complicated. And I think this is one of the reasons that the research that we do is trying to understand this in a much more simple genetic system. So as you can see, these mutations, particularly the ones up here, are kind of spread throughout the protein. And for us, this leads to some really fundamental questions that we would really like to answer. The first one is like, which target genes are affected? I told you that KDM5C regulates transcription. What does it regulate the transcription of? Do all of these different mutations fundamentally cause changes to transcription of the same genes? Or in fact, are these different changes causing different downstream consequences? We also have this really fundamental question, like the most characterized function of KDM5 is this demethylase activity, this removing of methyl marks. But you can see, I told you that the Jumanji C domain and this domain, this region down here, are the regions that carry out that enzymatic function. But you can also see there's lots of other changes that are nowhere near this. So is everything ultimately affecting this enzymatic activity, or is it more complicated than that? And I'm going to tell you the answer right now, and you'll be convinced by the end of my talk, and the answer is, it is more complicated than just this. And of course, which cell types are affected and what actually happens to them.
Okay, so this is where we're selling on Drosophila as a model system. So obviously all of our work is based on patience, right? Without patience, there is nothing, right? So it's difficult. Firstly, working on anything to do with the brain function, this is possessed. Having the mics on you?
Yeah, no, but you can tell. You've been projecting a lot.
Okay. So one of the things that's really difficult about like anything to do with neurons is it's in the brain, right? It's not easy to get to the brain, especially in an intact person. It's also, as we talked about actually this morning in that little chat, there's diverse genetic backgrounds, right? Every different patient has a different kind of genetic composition, and this actually makes the interpretation complicated. Like, how do you know which of those different clinical outcomes is due to the mutation in KDM5C? And of course, you can't test things in humans to start with. You need to start somewhere else. So I'm actually not going to talk about the mouse studies at all. If you're at all interested, Dr. Shigeki Iwazi is up the back there. He would love to talk to anyone about the studies that he's been doing with KDM5C in mice. What I'm going to tell you about is our studies using Drosophila. And I'm going to tell you that Drosophila has been used for more than 100 years now to unlock basic biology questions, right? And the reason that Drosophila is such a powerful system is that 75% of the genes in humans that actually have a clinical disorder are actually also found in flies. That flies have a gene that looks the same as that gene in humans. So just for comparison's sake, this is human brain, little fly brain down here. So despite flies not really looking much like a human, really being the same size as a human, actually the genes that flies have are highly conserved with what humans have. And for us, one of the advantages of being able to use flies is that they have this amazing toolkit of genetics that we can use. So in my lab, we actually have generated a fly strain in which we have completely eliminated the KDM. There's only one KDM5 gene in flies also, which makes it genetically simpler, which is why in some of my slides you'll lose the KDM5C part here and it's just called KDM5. So we have generated a fly strain that doesn't have any KDM5. We've also now we've taken those patient-associated mutations. Flies have the KDM5 gene, and we've made those same mutations in those fly strains. Okay, so now we're able to look at what happens to gene expression and everything else as a consequence to those particular changes to KDM5. We also have a different fly strain in which we've just taken away the enzymatic activity. Okay, so this enables us to kind of tease apart which of these effects are due to loss of enzymatic activity versus other things as well. And using flies, we're able to do cool genetic trickery to look at where KDM5 is binding in the genome and which genes it's affecting. We can look at we can look at what's happening to the axons of the neurons. And we can also do some cognitive assays on flies, which I'll touch upon in a minute. Many people are surprised to realize that flies have learning and memory, and all the same genes and proteins that are required in flies are the ones that we need to learn and remember. Okay, so we think that the power of this is our ability to kind of combine these things and ask these same questions that I was talking about initially. Like, what are the changes to gene expression that occur? Do they form a particular pathway that could possibly be dysregulated here? What happens to the neurons? Okay, so I'm going to tell you two mini stories that really emphasize the fact that it's complicated and that some of the things that KDM5 is doing in neurons is actually totally dependent on this enzymatic function. And then some of the things that it's doing are totally independent of its enzymatic function. And I think this really changes the way that we think about the clinical outcomes that appear from this. Okay, so I'm going to tell you, so this is actually what a fly looks like from front on. And I obviously know the green parts here, I've drawn in the green parts here. You're going to see some pictures. These are actually some neurons in the adult brain these are required for adult learning and memory of flies. It's called a mushroom body. It apparently looks like a mushroom. Actually, Hayden has a really pretty picture that makes it seem like it looks like a mushroom. But these are called the mushroom body neurons. And there's no direct equivalent to that in the human brain. It actually forms the function of several different regions of the human brain. Nevertheless, the way these develop is actually really similar to what happens in humans. So as I said, this is going to be 2 little vignettes, a tale of dimethylase dependent and independent functions of KDM5. So I'm going to tell you, the first story was carried out by a former PhD student, Samira Zamarat, who's now a postdoc elsewhere in New York. I'm going to tell you about one particular allele that she generated. So we have a dimethylase dead strain, called this 2-1GC, and the particular mutation that she was looking at was just outside this region of the protein. And so what she did was look in her fly strain that had this mutation, and she looked at what genes were being affected. So she looked at every single gene in the genome and asked whether it was up or down regulated compared to a typical fly. And the only thing that you need to know about this graph is that every single dot on here, many of which you cannot actually distinguish because they're in the middle here, every single dot is a gene. There are about 20,000 of these in Drosophila, and these are the 2 different genotypes that we have here. So the 2 different fly strains, this lacks demethylase activity, and this is the other mutation that we have. And if these 2 things were basically the same, then all of the dots should lie along this red line. And in fact, that's exactly what we see. So what we see is that this particular allele that's just changing 1 amino acid in KDM5 actually is effectively behaving exactly the same as a strain that just lacks enzymatic activity. And what we find when we look at those genes that are actually affected is that we find that actually transcription of ribosomal protein genes are by far the most predominantly affected here. So our original model was that KDM5 is regulating these genes in both the translation, and this is ultimately affecting neurons. So one of the reasons that we were actually really excited about this result is the translation. So translation is the process by which you take a gene that makes RNA, and then that gets translated into protein. Okay, so one of the things, the kind of funky thing about axons and neurons is that they actually make their protein close to where it's required. So if you've got a synapse here, so you've got axons, neurons talking to each other, a lot of proteins actually produce right here. And actually translation right there is actually really important. And in fact, translation is affected in other diseases that cause cognitive issues. For example, the most predominant cause of X-linked intellectual disability is mutations in the gene called FMR1. Mutations in this gene actually affect translation. There are other cases where translation is affected. And in fact, more recent data suggests that other diseases that are not intellectual disabilities, but they're things that degenerative diseases that happen late in life also affect translation. And so one of the things that my lab is trying to do right now is trying to restore translation in our fly model of this particular allele of KDM5C and ask whether we can restore cognition. Sorry, I was a little bit ahead of myself there. One of the things that I was going to tell you before that is that what we've done is in flies, we've tested the ability of this particular fly strain to learn and remember. So the way that we do this in flies is that we do an olfactory learning and memory test. They have a short-term memory, they have a long-term memory. The time scale is different to us. For a fly, 5 minutes is short-term memory and 9 hours is long-term memory. Whereas for us, it would be a different time scale, but this is what we do in flies. And we can see here that both dimethylase dead, a strain that lacks demethylase activity, and their other mutation strain, both have the defect in both short-term memory and long-term memory. So now what we're trying to address is we saw this change to translation. Maybe if we can restore translation here, we can then restore the ability of these flies to learn and remember. So the story that started with Samira is actually when Hayden also came to my lab. And the first thing that Hayden did was actually ask the very simple question of, you see these changes in cognition, maybe there's something structurally different about these fly strains in terms of their neuronal architecture. And what he showed very clearly was that in fact, this is a typically developing fly brain. You can see, I think my picture is actually pretty accurate here. You can see these 2 kind of L-shaped bundles of axons. And so these are the mushroom body. You can see that our mutation strain right here actually looks perfectly normal, right? So even though they have a learning and memory deficit, their neurons look normal. So we would argue that this is probably like a functional change in those neurons, possibly related to the change in translation. But actually one of the really interesting observations that Hayden then made was that he started to look at some of our other fly strains that have mutations in other regions of KDM5C. And what we find here is, so this is the one that I just showed you, where structurally they look really normal. So when we look at this fly strain down here, so we now start to see changes. And specifically what we see is that the axons don't either make the right turn and go the right direction, or they give the right direction and they grow too far. So whereas you would, these axons would normally stop right here and there would be a gap here, what they do here is they continue on across the midline here. And you also notice that up here, there should be a nice thick bundle of axons, and some of them is actually misdirected here. We also see that in a different mutation, all the way, another allele that's also all the way down here in a region of the protein that we have no idea what it does. These animals also have a change to the way that the neurons progress in the mushroom bunny. So instead of forming this nice L shape, they actually wrap around, and so they're misdirecting their axons here. So what's actually really interesting about this is that when we look at these two mutations here, these are actually much more similar to what we see when we just take away all KDM5 in those neurons. We see these kind of axonal guidance changes, and we also see this kind of crossing of the midline of the axons not knowing where to stop here as well. So because we do not see these changes either in this fly strain or in a strain that specifically lacks demethylase activity, these changes here are by definition totally independent of the enzymatic activity of KDM5. One of the other things that is really notable about these changes that we see is that FMR1 is, like I said, the most common form of inherited X-linked intellectual disability in humans. The fly model of this also shows the same thing, right? It shows these axons not really knowing where to stop. And in fact, Fragile X in flies, the characterization of this specific phenotype was actually linked to the realization that a very specific pathway was affected. This went on to be replicated in mouse studies, and actually then went on to the very first clinical trial of a drug to treat Fragile X kids. So what we're hoping is that our observations in flies will ultimately lead to further discoveries in things that don't fly. Okay, so I'm gonna sum up here and tell you that the very little bit of our research about KDM5 that I've told you about today is that we think that KDM5C is complicated. You know, it has this enzymatic activity, and we do think that enzymatic activity is important. I've shown you that actually, if you lose that enzymatic activity, you have deficits in learning and memory. And we also know that there's problems in synapses as well. And this is in the neuromuscular junction. That's Helen Balakazar's work, and I'll advertise her on the next slide. We also know that there's something else going on as well, right? Most of the rest of the protein is not related to this enzymatic function. And we think that this is also contributing to what KDM5 does in neurons. And that it's the combination of these things that actually help with normal cognition. And that disrupting one or both of these processes could also impact whether the severity of the cognitive deficits that occur in patients as well. So I'd like to end by just saying that, like, from our perspective, what we want to do is we want to understand the real basic biology of KDM5. We want to understand what is it normally doing during development, what goes wrong in different alleles that are found in patients, and we see this as like the fundamental first step in being able to identify different pathways that are affected, and ultimately that's going to lead to treatment for patients. So obviously this work wouldn't happen without a wonderful group of people in my lab. I talked about the work done by Hayden Hatch. I'll also advertise the fact that Hayden is presenting a poster around Main Street. Also Helen, she's done some really nice work on the neuromuscular junction. And also Mike, who we talked a little bit this morning about hormonal changes caused by KDM5C, and actually Mike's project has gone down this road in slides. Also, obviously, we need money to be able to do all of the wonderful things that we do. And I'd like to specifically note that way back, I think 2016, this project was really kickstarted by a pilot project from the IDDRC. And really was that little bit of money that enabled us to get started, and that led us down the road to getting other funding, which really enabled us to take off with this work. And I don't think we're going to do questions or anything, right? We're just going to go straight on to the... So I'm going to lead straight now onto the Rebel family. It's wonderful to meet you already. Hayden's going to do a little bit of an interview, and I'm going to leave it to Hayden to introduce himself and to introduce you guys.